We need to get the mRNA, we need to get the copy, we need to get the gene, but not the gene, but we need to copy the gene. It's like if you're at my house and I have a recipe book, and you see this recipe that's in the book, and you can't stay at my house and make it, you gotta leave, and you ask, can I take your recipe book home with me? No, you can't have my recipe book. So what can you do instead? You can make a copy of my recipe and you can take it to your house and make it. That's pretty much what's happening here. You can't get the DNA out of the nucleus. You can't remove the recipes. So a copy of the recipe needs to be made. That's what mRNA is. mRNA is a copy of the recipe. It's a copy of the gene. All right? Then that's going to leave the nucleus. It's going to go into the cytoplasm. So now we have the recipe. We need to read it. What's going to read the recipe? What's going to read the code of the messenger RNA? Ribosomal RNA. And you guys already know what that is. Because lecture C taught you guys about ribosomes, right? A ribosome is made up of what? Two main things. RNA and, starts with a P, rhymes with protein. Protein, right? A ribosome is protein and RNA. And it's ribosomal RNA. And the job of ribosomal RNA, by the way, where are most ribosomes located? Most of them. On your ER, right? Endoplasmic reticulum, rough endoplasmic reticulum. Why is it rough? Because it's got all those little ribosomes on it. It looks rough. The other ones kind of float around in the cytoplasm, correct? There's free ribosomes, and then there are those that are on the Golgi, or I'm sorry, on the endoplasmic reticulum. It's those ribosomes that are going to help us make protein. What are they going to do for us? They're going to read the code. They're going to read the recipe. Now we need a chef. What's the chef? That's the tRNA. The tRNA is going to construct the protein. What are proteins made up of again? What's a protein made up? What are the monomers of a protein? Amino acids. So the tRNA is going to bring along specific amino acids based on the code, which is being read by the rRNA. We're going to draw this, don't worry. And we're going to see an animation, two animations on this that are already on file. All right? So now we have the players in place. We have the gene, we have mRNA, we have RNA, we have tRNA. Four things in place to make a protein. Yes, sir?
Could you repeat your analogy? You said the transfer RNA was the chef, right?
That's the chef.
Okay, what was the rRNA?
That's the person reading the recipe. Okay? That's the grunt in the kitchen. Okay? That's the intern in the kitchen. And then the mRNA is the recipe. All right? The copy of the recipe. All right? So let's make protein. So we're going to draw a nice picture showing everything that I just said. And then we're going to see two animations on it, and hopefully by the time it's all over and said and done, in about 10 minutes, 15 minutes, it'll be crystal clear. All right? So I'm going to draw a cell. So that's a cell. Inside of that cell is a nucleus. Inside of that nucleus is DNA. So here's our DNA. We're going to make a nice short little gene. We have our two strands here of DNA made up of a gene, genes I should say. I'm just going to make it, I don't know, C, T, A, G, A. Let's go with C again. So that's CTA, GAC. All right? There's our recipe. We're going to copy it. But it's not going to be a direct copy, it's going to be what's called a complementary copy. So there's an enzyme that comes into play. You don't need to know the name of the enzyme. I don't care. If you take a biochemistry class, you're going to be going over this in such excruciating detail. This will look like baby stuff, and we'll get into all that other stuff. I only care about the complement, all right? So we're going to complement those bases of those nucleotides. So what the enzyme does is it grabs nucleotides, and it's going to stick them together. All right? The nucleotide with which base is going to be the first one that that enzyme grabs. What is it going to have? What as the base? So C is our base in the gene. What's going to be the base of the first mRNA, nucleotide of the mRNA? G, right? The C and G pair. So a nucleotide with a G is going to be grabbed. And then another nucleotide will be grabbed by the RNase, or by this enzyme. You don't know the name of it. What's the next one? A. Now, A is the next base, nitrogen base here. Now, if we had DNA as the other strand, what would pair with it? It would be, what pairs with A? T, if it was DNA. But we're making RNA. And with RNA, RNA does not have thymine. The thymine is replaced by another nitrogen base called uracil, all right? So instead of a T, we're gonna get a nucleotide with a U. So if you have an A on a gene, and you're transcribing that gene, and right now, by the way, we're talking about transcription. Transcription is the process by which we copy a gene. That's what we're talking about right now. In order to make a protein, transcription followed by translation. These are things I'm sure you guys have heard of. I'm just repeating stuff you already know. All right? But hopefully make it get nice and clear. G, what's the next one? What should that be? C, next one, that's an A, so this should be a U, and then the last one, G. There's our mRNA. We just copied the recipe. It's a complementary copy, but it's still a copy. It's still the same code. So it's a strand made up of all these nucleotides with all of these nitrogen bases. So we're going from here to here. And we can name that number 1. So the first thing that happens, number 1, transcription. So transcription is copying the gene. You transcribe a gene. Okay? Transcription is the noun, transcribe is the verb. Okay? So we've made this copy. But the copy can't stay in the nucleus because the machinery to make the protein ultimately does not reside in the nucleus, it resides in the cytoplasm. So how are we going to get it in the cytoplasm? We don't care, but we just have to know that it does go into the cytoplasm. So into the cytoplasm it goes. All right, so it's transported through the nuclear pores. You guys know what nuclear pores are, little holes in the nucleus basically. Now we have our mRNA here. Now it's in the cytoplasm. And it's G, A, U, C, U, G. Again, that's the code to make the protein. Now, when it comes to this mRNA molecule, we can even call that 2 if you want. This mRNA molecule, the codes are called something specific. So we have these 3 nucleotides right in order. We have these nitrogen bases, 3 of them in order. 3 of those nitrogen bases in sequence of an mRNA molecule is called a codon. For lack of a better, again, that's the code. As soon as you see the word, as soon as you hear the word codon, you know we're talking about mRNA. Codons do not exist within a gene. Codons only exist in mRNA. End of story. Okay? Something else that I want to point out. If you see a sequence of nitrogen bases on an exam, or you hear a sequence of nitrogen bases wherever, and T is mentioned, thymine is mentioned, immediately you know that's DNA. Immediately you know that it's not RNA. There is no thymine in RNA. If a uracil, if a U is mentioned on the exam, or you hear uracil, you know immediately it's RNA and that it's not DNA. You know it's not a gene. Okay? U only exists within RNA. T only exists within DNA. Cytosine, guanine, and adenine exist in both, but thymine and uracil are distinct. Thymine, DNA, uracil, RNA. Okay? Good to know. I promise you that it's good to know. All right? So now we have this mRNA, it's sitting in the cytoplasm, and now we're going to translate it. All right? So you transcribe a gene, you translate mRNA. Transcription happens in the nucleus, Translation happens in the cytoplasm. How are we going to translate this mRNA? How are we going to translate the code? How are we going to translate the recipe? RNA is going to read it first, all right? So, in order to depict RNA, I'm going to make it a different color. I'm just going to draw a dotted glob around that mRNA. I want you to think of it as my finger is mRNA and my hand is just going to kind of wrap around the mRNA. And that's kind of what happens, all right? That's going to allow the rRNA to read it, okay? So the blue thing that I just drew around the mRNA is rRNA. That's its job. It's to read the code. It's to read the codons. It's to announce it to the tRNA, which reside within the cytoplasm as well. So we're going to draw tRNAs now. We're going to draw them with little chicken feet. Those are tRNAs. They too are made up of nucleotides. So they too are going to have nitrogen bases. And there are going to be very specific tRNAs. And so, GAU is our first codon. So there exists within the cytoplasm a complement to that. Tell me what the complement to GAU would be on this tRNA, this first one right here. What should this first one be right here? The nucleotide should have what nitrogen base on it? If this is a G, this has to be C. The next one would be U, and then the last one, based on U being a nitrogen base on the mRNA, that would be at A. See that? Tell me what the next tRNA would be based on CUG. So this would be G, next one would be A, the next one would be C. All right? Those have a special name as well. Those are complementing the codon. Those are, for lack of a better name, called anticodons. Now I told you that the tRNA is responsible for constructing the protein, putting amino acids together. So they're going to be carrying amino acids. So let's put amino acids on the tRNA. That's an amino acid. That's an amino acid. And they're going to be different amino acids. Why? Because the anticodon is different. The anticodon is specific for a specific amino acid. So I'll draw them there. I'll put stripes in this one and I'll put little dots in that one. But both of those are amino acids. The red thing, I should say things because there's two of them. The red thing is tRNA. All right? And we'll just point at the little circles there. Those are amino acids being carried. Where are they carrying them to? They're carrying them to the messenger RNA. That's their destination. That's, in essence, what translation is. So we can put a third arrow here. These two guys, or gals, things, are going to literally, like a spaceship, dock. Again, the bases, the pairing, fit together like two pieces of a puzzle. So, this C right here is going to connect beautifully with this G. And this A right here, or this U I should say right there, is going to pair beautifully with that A. And the A is going to pair beautifully with the U. And it just sits right there. And it interconnects. And so now we have amino acids sitting right here. And then the next tRNA right next to it docks. Now these two amino acids are right next to each other. What are they going to do? They're going to be bound to each other. What have we just produced? What kind of structure have we just produced? It starts with a P.
Protein.
It is protein structure, but specific. There's 4 types of protein structure. What are we producing here? We're putting the amino acids in a sequence. Primary structure. What we're doing here, by lining up these amino acids next to each other, based on the anticodons of the tRNA, which are based on the codons of the mRNA, which are based on the gene of a DNA molecule, the original recipe, is to stick amino acids next to each other so that we can, at some point, do that. So we talked initially about primary structure of a protein. That's how it's done. and the amino acids are put in the correct